Morning, everyone. Thank you for joining us this morning in our endotoxin detection webinar. Before Jiya gets started, I just wanted to inform you all that the Q&A has been enabled, so you can ask your questions there. If we do have some time, Jiya can address it, or we can address it offline as well. So basically, this webinar provides opportunity to learn more about endotoxins and the regulatory requirements that we have with endotoxin testing, and obviously the full set of solutions that we offer from LONZA. So this is the methods, the reagents, and the solutions that we have. So today's speaker is Jiya. Jiya John is our application specialist, a senior application specialist at Whitehead Scientific. She has been at the company for 5 years now, and she has a strong background in cell and molecular biology. Her primary role basically focuses on technical training, troubleshooting, and customer relationships. And she's very experienced in the endotoxin workflow, including installations, IOPQ validations, and troubleshooting. So she has prepared a lovely presentation for all of you today. So yeah, without further ado, I'll hand over to Jiya, and I hope you enjoy. Thank you.
Thank you, Felicia. I'm going to share my screen. Let me know if you can see it.
Yes, we see it.
All right, thank you. Thank you guys for joining our webinar titled Endotoxin Detection, the Key to Safe Pharmaceutical Products. And thank you for the lovely introduction, Anisha. And you basically did highlight, the agenda for today's topic. So we just want to give you a brief overview of, what is endotoxin, what are the regulatory requirements, what are they talking about? When you say what do you need to do for endotoxin testing, there are different types of tests. We will just go through that and see which one would be the best. We will also introduce you to the alternative method that we are talking about these days in the endotoxin space, which is the alternative method using recombinant technologies. And then I will touch on the readers that we have from LONZA, the software that we use, and also just conclude with the complete testing solutions that LONZA offers for the specific testing. So to start off with, what is endotoxins? So endotoxins, they are biological molecules, also known as pyrogens. They are found in the outer cell wall of gram-negative bacteria. So these pyrogens, or these biological molecules, they are continuously being released into the environment during cell death and cell division. Some of you may have heard the term lipopolysaccharide. So that is basically the chemical name of endotoxin. You can use it interchangeably. It comes from the naming comes from the chemical structure of this molecule. So it has a polysaccharide chain and a lipid A part. So it is the lipid A part that is responsible, or the biologically active molecule that is responsible for the pyrogenicity of this pyrogen. And as you can see on the structure here, it has both the hydrophilic structure because of the lipid A region and a hydrophobic part. Because of this unique structure, you could come across cases where you are trying to detect endotoxin in certain products, and you might encounter some issues, and the reason behind that would be the structure of the endotoxin itself. So why is endotoxin a cause for concern? So endotoxin, it is a pyrogen, as I mentioned before. So what is a pyrogen? Pyrogen is basically a substance that can cause fever in humans and animals when present in the bloodstream. So you get pyrogen, there are different sources from where you can get pyrogens, and this would include your bacteria, virus, yeast, and mold. So what happens is that when you have your pyrogen entering your bloodstream, it interacts with your immune system and it causes some adverse reactions, including inflammation, fevers, chills, shock, and severe cases, in some cases, death. Pyrogens can be classified into two parts. One is your endotoxin and one is your non-endotoxin pyrogens. So endotoxins is specifically coming from the gram-negative bacteria, and the non-endotoxin pyrogens are coming from your gram-positive bacteria, yeast, mold, and viruses. So endotoxin specifically is a cause for concern because it is the most potent type of pyrogen. An example of a gram-negative bacteria would be your E. coli. So as we know, E. coli is found everywhere, outside, even within our system. So it is found everywhere. And one of the main challenges that we have with endotoxin is that it is stable under extreme conditions. So it is resistant to heat or chemical, chemical destructive methods. So autoclaving and trying to get rid of these things in your lab is something very challenging. This is a most potent type of pyrogen that you will most likely encounter in your manufacturing processes. So it becomes a significant challenge in the manufacturing of parenteral drugs and medical devices. So in biochemistry, we always have that saying where we say all enzymes are proteins, but all proteins are not enzymes. So the same thing applies here, where we say while all endotoxins are pyrogens, not all pyrogens are endotoxins. So if you want to test for pyrogen itself, there is a different test kit that we have, which is called the PyroCell MAT system. That is something we do provide from LONZA. This is not being covered in this webinar, but if you do require further information for this particular kit, please do reach out to us. And just moving on to who needs to perform the endotoxin testing. So if you look at the patients that are with trauma or that are placed in the ICU, they are more vulnerable to the effects of endotoxin. Even small doses of endotoxin can cause fever in them. So the detection of endotoxin becomes vital in the pharmaceutical and medical industry to ensure patient safety and product quality. It is a mandatory requirement for manufacturers of injectable drugs, medical devices, dialysis products, and nuclear medicines to test for endotoxin in their products in order to minimize the amount of endotoxin present during all stages of manufacturing. Just to highlight, for researchers that are doing cell culture work, sometimes the presence of endotoxin can also affect the growth of cells in vitro. They can get invariable results from the different tests that they do. And the sources of endotoxin could be the use of recombinant proteins, cell culture media, FPS, or water. So for those labs that are doing research work, we do have research use only kit for testing the endotoxin. And this would be from our supplier, Highcult Biotech, where they have a chromogenic endpoint assay that you can make use of. So that is basically just a brief introduction on endotoxin and why we need to test for it. And now moving on to the regulatory requirements. What do the regulatory requirements say? So we have a harmonized bacterial endotoxin monograph that can be found in the three major pharmacopoeia, which is your US pharmacopoeia, European pharmacopoeia, and Japanese pharmacopoeia. So all these three, they have harmonized study details. So more or less what you read in each pharmacopoeia would be the same. Just to highlight, products may contain endotoxin. However, it should be within the allowed regulatory limit. So endotoxin is not something we can get rid of, but what the aim should be for us to keep it to the very minimal amount. So there are also some supporting documents that allows you to troubleshoot more or gain more specific knowledge or specific questions that you may have regarding the endotoxin testing. So for your parenteral drugs, they do have a guidance for industry Q&A. This document is available on your website. This is something you can download and have a read through. And for medical devices, they have a standard document that can also be applied when you are doing endotoxin testing with medical devices. A point to note is that the endotoxin kits that we have from LONZA. They are already approved by FDA. And the protocol is written in a way to comply with available regulations. So once you are using the LONZA endotoxin kit, you are already complying to details or the steps that you need to follow according to the pharmacopoeia. So the sterile parenteral drugs and medical devices must be tested with BET according to the pharmacopoeia, and it must be shown to contain less than the endotoxin release limit. So this value here, the endotoxin release limit, most often it is mentioned in the defined regulatory documents. In cases where you don't have this information, there is a formula that you can use to calculate the endotoxin release limit, and I will touch on that in the following slides. So the other requirement is the validation. So as we saw on the first slide, because of the structure of endotoxin, you may encounter issues with testing for endotoxin in specific products. So validating that you can use endotoxin testing for your specific product becomes very important. So to summarize, the three essentials when you are looking into starting endotoxin testing is first to establish the endotoxin limit of your pharmaceuticals and medical devices. Second would be to establish procedures for validating the use of endotoxin in the lab. And this can be both product independent and product dependent validation. So you want for product dependent, it would be to see whether there's any inhibition or enhancement assay from the endotoxin on the product. And lastly, it would be to establish procedures for conducting routine testing. So how do we calculate the endotoxin release limit of a product if we do not have information already? So for this, I need to introduce you to a terminology called threshold pyrogenic dose, or TPD. So TPD is basically how much endotoxin can we safely give a patient. And this would depend on the product type, which is either the injectable drug or medical device. Will the product come into contact with CSF, which is cerebrospinal fluid, because a lower endotoxin limit needs to be applied here. And in certain cases, time can also be a factor, because if you are giving repeated doses within an hour, time needs to be taken into consideration. So TPD for injectable drugs and medical devices, they are a fixed value, and these values are defined in the BET monograph as a constant K. And the definition is K is endotoxin units per kilogram of body weight that can be administered in 1 hour. So it's EU per kg per hour. So according to the BET monograph, the constant TPD K for injectable drugs that does not come into contact with CSF is 5 EU per kg per hour, whereas for the TPD for injectable drugs that comes in contact with CSF is 0.2 EU per kilogram per hour. And now for medical devices, they have defined their constant as EU per device. So the K for medical devices that does not come into contact with CSF is 20 EU per device. And the medical K for medical devices that come in contact with CSF is 2.15 EU per device. So you need the information on TPD in order to calculate the endotoxin release limit, because the formula for endotoxin release limit is K over M, where K is your threshold pyrogenic dose, and M is the maximum dose of the drug in units per kilogram per hour. And again, calculation of ERL will depend on the product type, whether it's injectable, intrathecal, or radiopharmaceuticals, or medical devices. So I have put two examples of how do you calculate the endotoxin release limit. So sometimes you have to take into consideration the drug potency, if that information is available. So then you will have to use the formula K over M times the drug potency to get to the ERL, endotoxin release limit. For medical devices, the formula is a bit different, where they take into consideration the number of devices that you have used. So with medical devices, it works a bit different. you can pool samples from a specific lot. So you can take maximum 10 devices from a batch. So that's why they take that into consideration. And you multiply the number of devices by the total extraction volume to get to the ERL value. And what are the three methods that are acceptable by the regulatory board for the endotoxin testing? That would be your rapid rabbit pyrogen test, which is something we have been using historically. Then we have the bacterial endotoxin test, which is what we will be discussing today, and then the monocyte activation test. As I mentioned, monocyte activation test is a test used to detect both endotoxin and non-endotoxin pyrogen. Rabbit pyrogen test is something that is being discontinued this year. So there is a plea for all the medical device manufacturers and injectables for them to actually move from the rabbit pyrogen test to either the LAL BET test or the monocyte activation test, depending on the aim objective of the product. So LAL test is something that is widely used in the parental drug and medical device industry. So I just want to introduce you to what is this BET or LAL test. So according to the pharmacopoeia, the definition of bacterial endotoxin test, it is a test used to detect or quantify endotoxins from gram-negative bacteria using amoebocyte lysate from the horseshoe crab, Limulus polyphemus or Tachyplis. So the naming convention comes from basically, you can either say bacterial endotoxin test or otherwise, most people also say Limulus LAL test, Limulus amoebocyte lysate. And this is because they're using the horseshoe crab, Limulus, and then the blood cells, amoebocytes are basically like the blood cells, and they're using the protein lysate from this blood cell. So that is where the alternative name of LAL comes from. So the history behind this is that there was two researchers, Frederick Bang and Jack Levin in 1960s, where they noticed that these horseshoe crabs, the blood cells of these horseshoe crabs, they form clots around the bacteria when they get attacked by the bacteria. They form clots around these bacteria, so in order to protect themselves, from their attack by releasing certain proteins. And then they studied this clot formation, adapted this to develop the commercial kits that we are seeing or we are currently using for endotoxin testing. So this is, I have just a picture of the horseshoe crabs, donating the blood. And it is interesting to see that the color of their blood is actually blue. So the principle behind the LAL assay or the BET test, so as I said, it's the blood cells from the horseshoe crab that we're using for the kit. So the lysate has a mixture of naturally occurring proteins, and the main protein or the key protein that is involved in the binding of endotoxin is your factor C protein. The factor C protein, if there is endotoxin present, it binds to the endotoxin and it becomes activated. So for those people who are already doing endotoxin in their lab, you may have noticed, there's a slight lag before you see some peaks coming up, especially with kinetic assays. Before the graph comes up, you would see a bit of a delay. And the reason behind that is that, the protein needs to be binding to the endotoxin and go from inactivated phase to activated phase. So that is the lag phase that you are seeing in your software. So once we have your activated factor C, it then goes and activates another protein, which is your factor B, which then converts a pro-cloting enzyme to a cloting enzyme. So for commercial purposes, they have, you have to somehow see this reaction happening. So then they have come up with a substrate that you can that can react with the enzyme and give us a detectable signal. And this signal can be in either the gel formation or turbidity measurement or yellow color. So I will just explain the three different kits that we have for the endotoxin testing. So first is your gel clot LAL assay. So this is one of the simplest form of the endotoxin kit that you can get. So basically here, you look for the formation of a gel. So if there is endotoxin present in the sample, a gel will be formed, and this experiment happens in a test tube. So a formation of a firm gel that stays intact momentarily when it's inverted indicates there is endotoxin present. This experiment gives you a qualitative readout, so it's a yes or no answer. But the sample setup or the experimental setup is very easy. You have your sample, you mix it with your LAL reagent, the lysate, in a test tube, in a glass test tube, incubate it in a dry heat bath at 37 degrees for 1 hour. Gel plot is something that would actually help you when you are doing some initial screening for new products. The advantage of this assay being it's simple and you do not really require expensive equipment. Disadvantages being is that it is subjective. So I can look at the test tube, I can look at the gel and be like, okay, that is a firm gel. Someone else can come and say, no, that does not look, firm to me. So that's where the subjectivity comes in. And it's qualitative. So if you want to know how much endotoxin is present, then this assay will not be the one to go for. And it is labor intensive. Just a point to note for people who are doing gel plot at the moment. The caution is that because the formation of gel that is happening in the test tube is due to electrostatic bonding, it is one of the weakest form of chemical bond. So we tell the people that are doing gel plot assay to make sure that where you are performing that assay, there shouldn't be any other instruments that can be like a source of vibration, like your centrifuges, your vortexes, etc. It should not be in the same bench where you are doing your gel plot assay because it can also disturb the formation of your gel. Now, the kinetic turbidimetric assay is also similar to your gel plot assay setup, where you have your instead of a gel formation, we are measuring the turbidity inside the sample. The good thing about or the benefit about this assay is that it's kinetic, so it reads as it happens, and the readout is quantitative. So the degree of turbidity is measured to determine the presence and amount of endotoxin present in your sample. And for this assay, you would need a 96-volt plate where the sample is added and an incubating absorbance reader because the sample has to be pre-incubated at 37 degrees for 10 minutes in this incubating absorbance reader before you add your lysate. So that is the difference between the normal gel plot assay and the kinetic assay. You would need an incubating reader and also a software to help you get to the calculation of how much amount of endotoxin I have in my sample. The advantage being it is quantitative over a large range. So you can get any reading between 0.01 to 100 EU per ml. The chromogenic LAL assay here, they instead of a coagulant where it turns into turbidimetric or gel formation, they actually added a synthetic chromogenic substrate which produces a yellow color. So here the protocol setup is exactly the same as for the kinetic turbidimetric assay, where you have your samples added to the 96-volt plate, pre-incubated at 37 degrees for 10 minutes, and then mixed with the lysate. If endotoxin is present, it will cleave the synthetic chromogenic substrate that is present and then releasing the yellow color, which will then you will see on the ELISA plate. The presence, you will see the yellow color forming. So this indicates the presence of endotoxin. The advantage, again, it is quantitative over a large range, which is 0.05 to 50 EU per ml. That is with standard curve range. Disadvantage is you would need an incubating absorbance reader and also a software to help you measure the endotoxin concentration. So which assay is the best? So the answer to that would actually depend on few points, depending on the workload and what is the nature of the sample you are testing, the budget that you have, the equipments that you have in your lab, and need to quantify your results. So with the gel clot assay, as I mentioned, it is labor intensive, but it is something that you can use for initial screening of your products. And the kinetic assays, a general trend is that you would do water samples for turbidimetric assay, and the biological products like your vaccines and antibiotics, you would use your chromogenic assays because it is more sensitive. Turbidimetric assay, the standard curve range starts from 0.01 EU, whereas the chromogenic assay starts from 0.005 EU per ml, so it is more sensitive. Before I introduce you to RFC assay, I just want to let you know that LONZA does have an assay selection guide. So even though you are aware of the different kits that we have available, you can download this selection guide. I think we will distribute this document after the webinar where you can answer a few questions, and then it will direct you based on your answers to the right kit that you need with the right sensitivity. There's also a webinar that LONZA has done, which also talks about which method is best for me. So on the LONZA webpage, they have this webinar available once you have registered on their portal. So this is also something I would like to direct you guys to. So before we talk about the RFC assay, which is the alternative method using recombinant technology, I just want to discuss the horseshoe crab itself, which is the hero of this endotoxin testing. So the horseshoe crab, the Latin name is Limulus polyphemus. So there's two species, one that you can find in the east coast of USA, the American region, and then you will find another species called Tachyplus in the Asian continent. So if you're working in a lab in the Asian side, you may actually come across the endotoxin kit saying TAL reagent instead of LAL reagent. It just indicates that they have sourced the lysate from the species from Asian side. So what is happening with the horseshoe crab? Horseshoe crab is a living fossil. Excuse me. It has been living for over 400 millions of years. So it does not have any natural predators. So it doesn't really had to undergo any much evolutionary changes. So it is, as from the get, it has not had to go through much changes. So it is why it is called a living fossil. For endotoxin testing, around 500,000 crabs are captured and blood every year. So manufacturers who are producing the endotoxin kits, they do make sure to release these horseshoe crab back safely into the environment. But studies have shown that, after they donate their blood, some level of mortality and some sublethal impacts, such as decreased reproductive fitness, can be seen post-bleeding. So this is something that we are seeing in the population of the horseshoe crab also. If you look at the horseshoe crab in the East Asian side, the population is basically extinct now. And the other reason that is also causing this decline in population is that horseshoe crab is something that they used to use as a bait for fishing, and there hasn't been much regulation happening in the Asian side. So in the American side, it's a bit more stricter. They are trying to conserve the population that they have at present, but they are raising concerns now over the welfare and continued use of this animal in the pharmaceutical industry. So globally now, there is a growing appeal for recombinant technologies to come and help alleviate and eventually replace the use of animal resources, which is our horseshoe crab. And this is where our recombinant factor C assay comes in play. It is an animal-free alternative kit, and studies have shown that it is comparable to the traditional lab assays. So LONZA was way ahead in their thinking. back in 2003 already, they had manufactured their first endotoxin kit based on this recombinant technology using recombinant factor C. However, it took more than 10 years for FDA to actually approve a drug based on the recombinant factor C testing method. And the drug that was approved is called Emagalti. It is a drug it is used for the prevention of migraine in adults. So more people are looking into our recombinant factor C as an alternative method now more than before. And fortunately, both US FDA and European Pharmacopoeia has now accepted RFC as an alternative method to traditional lab assays. If you are a lab that is currently doing traditional lab assays on your product and you want to validate the RFC assay, LONZA does have validation protocols available for you to try it out. So from the European Pharmacopoeia side, they have included this chapter as of July 2020 already. From the USP side, they have approved the inclusion of bacterial endotoxin testing using the recombinant technologies. They have the latest update on this can be found in, you can actually join a webinar that is actually coming up on the 1st of April by LONZA itself. It is a short 30-minute webinar on 1st of April. They're actually touching more on this recombinant factor C assay and the new updates regarding the chapter from the recombinant factor C on the USP Pharmacopoeia. So the principle of RFC assay, so compared to the traditional lab assay, so this is the enzyme cascade for the traditional lab assay where you have few proteins that are involved in the enzyme mechanism. Compared to that, RFC assay is actually a single enzymatic step. You have your recombinant factor C that they have cloned from the horseshoe crab. Once it attaches to your endotoxin, it becomes activated, and then it cleaves a fluorogenic substrate, giving off a fluorescence reading at the end of the assay. So the difference between the traditional lab assays and the RFC assay, one would be that it is quantitative. It's a quantitative endpoint assay. So it takes 1 hour. It's you have to do the reading at time 0, and then after 1 hour, you need to take a second reading. So it's an endpoint assay. And it does provide a strong detection signal compensating for the amplification cascade that we see with the traditional lab assay. The other thing I wanted to point out was is that with the traditional lab assay, there is an issue of glucan contamination coming into play because glucan is a contaminant that you can find in manufacturing processes. This glucan can interfere with the traditional lab assay, giving us false positive results. So moving to the recombinant factor C assay, this issue does not come into play because there is no pathway for them to interfere with this assay. So that is another advantage of moving over to the RFC assay. The disadvantage would be that since it's a fluorescence-based reading now, you would need a new fluorescence-based reader for conducting this experiment. So just to highlight the experimental workflow, it's very similar to the traditional assay, where you prepare your standard curve, you pre-incubate them in an incubating absorbance reader or fluorescence reader. And then the only place where there would be a little bit of a change in the way you prepare your sample is that with the traditional assay in step 4, you add your lysate into your samples. With the RFC assay, you have to do a little bit of a pre-prep, but the calculations and everything is already provided in the product insert. You just have to follow the product insert, the table that is found in the product insert, prepare a working reagent by mixing the substrate, buffer, and the recombinant enzyme, and then you add that to your sample, and then you start your assay. So that is the only place where you have you find a difference between the traditional assay and the RFC assay. So once you have set up your assay, you can let the software do the reading. First reading would be at time 0, and then the second reading would be at 60 minutes at 37 minutes. And the software would then calculate the difference between the 60-minute reading and the 0 time interval. And it also plots the standard curve for you and provides you information on the concentration of your unknown samples. So the benefits of RFC assay, it's a non-animal alternative. The second highlight that I want to bring out is that it doesn't have any biological variation. So traditional lab assays, because it's coming from a natural source, the lysate is coming from an animal source, you will have batch-to-batch variation that you will find. But with the RFC assay, that is not an issue anymore. There will be batch-to-batch consistency. There will be a security of supply. There's no more asking, oh, are we going to be will there be shortage of these kits, because now it has moved on to the recombinant technology. And the other highlight would be that studies have shown comparable results to your traditional lab assays. So talking about the reader that you would need for the RFC assay, if you're moving on to that, I just want to point out that the ELX 808 reader, which is the reader that most of the labs that are doing endotoxin testing uses, this reader is actually being discontinued now. It actually discontinued in 2023, and with service support ending in 2029. So most of the labs that are currently using ELX reader, they need to look for alternative readers. So this would be a right time to maybe looking at investing in a reader that can do both traditional lab assay, which is your absorbance-based reading, and then a fluorescence-based reading for you to move on to the RFC assay. So from LONZA, they have actually launched 2 modes of readers. One is your Nebula multi-mode reader, and then one is your absorbance reader. So the multi-mode reader allows you to do both traditional lab assay and the recombinant factor C assay, because it has both absorbance and fluorescence mode. This reader can also be used for math assay. And then for labs that are still, they just want to stick to the absorbance kits fully, they can look at the Nebula absorbance reader as a direct replacement to the ELX 808 to conduct the traditional lab assays. I would just like to also highlight, the investment that you put in for the WinKQCL software. So most of the time when we are talking to researchers, they're like, it's a big investment they need to have for setting up an endotoxin lab. And when it comes to software also, they're like, do we really need the software? So I just want to highlight, the importance of having the WinKQCL software for your endotoxin analysis. So WinKQCL software, it is designed and developed specifically for the purpose of endotoxin testing. It is a fully integrated 21 CFR Part 11 compliant solution. It can do quantitative endotoxin and detection analysis, data management, reporting, and training. So basically, if you have the endotoxin kits from LONZA and using the WinKQCL software, you are compliant with the pharmacopoeia. So that is a very big plus point for labs that have to be undergoing audits and for QC purposes. So the WinKQCL software, what it does, it calculates the amount of endotoxin asphalt in your sample, and it also gives you the value of PPC recovery, PPC concentration, which are all important information that you need to validate the product suitability for endotoxin testing. So once you purchase this WinKQCL software, it can be used for both your traditional lab assays. It has a module for your RFC assay and math assay. It also has preset templates for routine testing, initial qualification assay, and the inhibition and enhancement assay. Other highlights that I want to bring out is that the software itself, it has reader-specific validations to make sure that the instrument itself is working properly. The reader can be interfaced, the software can be interfaced with multiple readers in multiple labs around the world, all storing data in a single database. So if you have a company, you have branches in different parts in the country, all of you can do experiments and all the information can be stored in one single database. So the head office can basically have access to all the data, all the results for each area of your company. Trending is another feature that I would like to highlight. So trending is advantageous so that sometimes you come to a point where you don't know what is causing the result that you're seeing. Now you're seeing inconsistent results. Is it because of the kits that you're using? Is it because of the accessories, the plates or the reader? So in order for you to pinpoint to a reason behind why you're seeing certain results, you can actually use the trending feature on the software. And then the other highlight would be that you cannot start a run if you are using expired kits, expired reagents or consumables. So this is a big plus point from a QC point of view. Then the software can also then be integrated into automation workflow. So something for future, if you are looking at expanding your lab, you're getting a lot of samples and you want to automate your workflow, the same software can then be just integrated into the automation workflow. And another highlight would be that the software can be connected to LIMS to export your data. And then this is the screenshot of our WinKQCL software. They have the templates, endotoxin test, all the tabs that is required. But the one thing I want to highlight, most people miss to see is that right at the end, there is a calculator button where you can actually input the values and then the calculator will calculate the dilution factor, what is the volume you need, what is the volume you need for maximum valid dilution. They can calculate the endotoxin release limit and stuff. So please make use of this calculator button if you already have the software with you. So with that, I would like to summarize the talk by saying from LONZA, if you are starting your endotoxin lab and you are going with LONZA, they do provide a complete testing solution for your endotoxin workflow. They have the traditional bacterial endotoxin test. They have the MAT assay for your pyrogen testing. They have the recombinant assay test. And now they have the instrument also, which is your nebula reader. They have your software. They also have your accessories. So on the accessory point, I just want to highlight the accessories that we have. And just to mention that the two things that you need to look out for when you're buying accessories for your endotoxin testing, that it has to be less than the limit of detection of the kit that you're using. So if you're using a kinetic assay, chromogenic assay, the limit is 0.005. So the accessories that you use have to have endotoxin concentration below that limit. And the second test that LONZA does is that it makes sure that the endotoxin accessory that you're using for the endotoxin testing is suitable, because remember, endotoxin, it has the hydrophilic and hydrophobic nature. So sometimes the endotoxin can bind to your consumables. So it is important to make sure that the consumables that you're using are suitable for the test that you're doing. So LONZA does both these tests and make sure that the consumables that you use are suitable for your work. And just lastly, to point out that LONZA does have a QC Insider portal, which you can register online, and they have various resources like webinars, seminars, presentations, the assay selection guide that I was mentioning about that you can download at your time. And they also have an e-learning module. So they have some few online training modules which will help you understand endotoxin a bit more in detail and become sort of like an expert in the field that you are working. So you can either register for all the different modules, or you can choose the module that is applicable to you. And then at the end of the online training module, you actually get a certificate of completion. Right now, they actually have 2 modules that are free of charge, free modules. So they have the recombinant factor C and the MAT assays that once you have registered, you can access them free of charge. And just note that once you have registered, you have to finish the course within 6 months for a free module. And for the ones with a part number, you have to finish the course within 1 year. And with that, I say thank you. Any questions?
Yeah, I don't think anyone has questions right now. But if you do think of anything that you want to ask, you're welcome to send Jiya an email or myself, and we will attend to it as soon as we can. This has been recorded, so the recording will be available for you afterwards as well. And let's just give it a little while more. I don't think I can see anyone's hand raised. No questions in the Q&A. Yeah, so if that's it, then I think we can close the session. Thank you all for attending and listening to Jiya. Just reach out if you have anything that you'd like to ask. Yeah, have a great day further. Thanks, everyone.